Microglia make up roughly 10% of the cells in your brain, and they're not neurons. They're resident immune cells, and they spend most of their time in a surveillance state, extending and retracting fine processes to monitor the chemical environment around the neurons, clearing debris, pruning unused synapses, and supporting the health of the cells around them. Hi, I'm Heather, and I build longevity protocols where biological system science meets the source code of reality. When microglia detect a threat, they shift into an activated state and release inflammatory cytokines like IL-6 and TNF alpha, reactive oxygen species, and proteolytic enzymes designed to destroy pathogens and clear damaged tissue. That response is supposed to be temporary. The problem is that microglia can get stuck in the activated state, and the very molecules they produce to protect the brain begin damaging the neurons they were supposed to be defending. PET imaging using translocator protein ligands has documented sustained microglial activation in roughly 80% of Alzheimer's patients and a significant proportion of Parkinson's, ALS, and post infectious cognitive decline cases. The activation often precedes clinical disease by years, which means it is upstream of the pathology that eventually shows up on a clinical workup rather than downstream of it. What shifts microglia from protective to destructive is the sustained presence of inflammatory signals. Insufficient sleep dropping below 6.5 hours, chronic psychological stress, heyo, blood sugar volatility, gut permeability sending lipopolysaccharides across the blood-brain barrier, and environmental toxin exposure. Every one of those inputs is modifiable through specific interventions. 7 to 9 hours of consolidated sleep, a diet that holds postprandial glucose within 40 milligrams per deciliter of fasting baseline, and active gut barrier support through fermented foods and polyphenol-rich plants address the three primary inputs that keep microglia in surveillance rather than attack mode.